Okay, this is the recording. There will probably be a couple of them at least for lecture 11 prep. We're going to be looking at pedigrees. So with human traits, we're definitely not going to be looking for ratios. The number of progeny is so small. And we certainly can't be doing crosses and that sort of thing. I mean, besides the obvious ethical reasons why you wouldn't do experiments with people, it wouldn't work anyway. What we have to do instead is wait for a family to do the experiment for us and then work backwards from the results and figure out how the trait is segregating. So the main purpose of pedigrees is to figure out how a particular trait is being passed on. Is it on the autosomes or the X chromosome? Every now and then there is a Y-linked trait, which is interesting. They're super rare because there are not very many genes on the Y chromosome. And also whether it's a dominant or recessive trait. If you're in a family and in that family there is a particular genetic condition segregating, and you are thinking of having children and you're worried about this, if you find out it's dominant, a dominant allele, and you're not showing any symptoms and you don't have it, you're okay, right? So this kind of information can really help people who are trying to make decisions, important, life decisions. Okay, so as I said, we can't do crosses. We use information from pedigrees, which look just like family trees, except we have special symbols that we use. And we use this, as I said, for single gene traits. There's lots of things we're really interested in, especially things like psychiatric conditions or tendency to heart conditions and that sort of thing that are affected by multiple genes and even environmental influences. So we're really focusing just on those conditions that are caused by a single allele of a single gene, right? And so we also I want to make sure it's clear that we're interpreting the pedigree information based on what we have. If we only have, one generation and one affected child, we can't know for sure. Sometimes we do, but a lot of the times we can't be sure what the mode of segregation is. So we select the most likely one based on making the fewest assumptions, which I'll explain in a minute. And if we get new information, it is possible to then change our interpretation. We update it because now we have different information. And that's how all of science works. We never get hopefully too stuck into our way of thinking, because new information will help us modify and make better and more accurate our interpretation. So if we're thinking something looks like it's dominant for a couple of generations, and then the next thing you know, here's a case where we can see it's clearly recessive, it's okay that we change our minds. And if I have in the past, I don't know if I'll do it this time, but I have in the past had on midterm a question that says, tell me, here's this pedigree, tell me what mode of segregation you think it is, and explain why, because I'm mostly looking for correct reasoning. And then, okay, here's like 10 years later, here's some more children born. It looks a bit different now. Tell me how you would change your interpretation and explain why. What did the new information tell you? And that's just to get you used to thinking, like, it's not about getting the correct one. It's we choose the most likely one based on whatever, even if the information is limited. But then when we get new data, new information, we either get more confident in our first interpretation, or we say, we didn't know that, so now we have to change the way we interpret it, right? So just keep that in your mind. Okay, so when we look at the pedigree, we can figure out the mode of inheritance. Is it a dominant allele? Is it recessive allele that leads to it? Is it an X-linked trait? If it is, we're happy because we only have to look for the causative gene, the gene that's responsible on one chromosome, versus autosomal. With 22 chromosomes we have to look through to find the actual gene. So weird. You can also sometimes figure out what different people's genotypes are. If it was an a dominant condition, for example, and a person didn't have it, we would know they were homozygous for the recessive allele. And that could be really helpful information for them if they're thinking about planning a family and so on. Now, in the next lecture, we'll be talking about probability, and we'll be talking about using probability calculations to figure out the chance of a particular individual in a family having a particular condition or being a carrier, heterozygous, for the condition and so on. But that stuff we'll be talking about in lecture 12. Okay, and I really want to be clear about this because it's super important. You cannot use ratios in your reasoning. You know, there are 4 kids and one of them has the condition, oh, it's a 3-to-1 ratio, it must be autosomal recessive. No, not necessarily. There are cases where 2 parents are heterozygous and they have 4 kids in a row and they all have the condition, and that's that's 100% affected individuals. The families are way too small for us to see any meaningful ratios. And especially with families that only have 2 or 3 kids, there's no way you're going to see any kind of ratio anyway. We'll be focusing on ratios a lot when, we cross 2 mice and we get 35 babies over I don't know how many litters, and here's the ratio, and tell me how the gene works. But we don't do that with human genetics because the numbers are just way too small. Okay. And the other thing, the traits are usually quite rare, not always. Cystic fibrosis is a notable exception, but the traits tend to be, the conditions tend to be, like 1 in 100,000 people, 1 in 50,000 people will have them. And that means it's very unlikely that people marrying into a family are heterozygous, are carriers for the condition. And so what we do is we make assumptions. This is just kind of our conservative rule for analyzing our data. We're assuming that anyone coming into the family, marrying in, like the spouses of siblings and so on, we're going to assume that they're homozygous for the normal allele unless there's direct evidence, right? If it's a dominant condition and they're affected, obviously they're not normal, right? Homozygous normal. They obviously have the condition. And if they're not affected and their spouse is not affected, but a child is affected, they must have been a carrier, right, under some conditions. So you basically don't assume that anyone's a carrier unless you've got some evidence. Whenever you make a claim, you back it with some kind of supporting evidence. And so that means we're really conservative in how we interpret, and we choose the mode of segregation that requires the least number of assumptions. Okay, and you'll see some, I'll talk about that as I talk through some of the examples. Okay, so just like any scientific area, topic, or whatever, we have a language, and so you'll notice there's lots of new terms to learn and understand, and there's also, when we look at pedigrees, symbols to learn and understand. I know most of you will have had this in a previous class, but I'll just go through it. So males are shown as squares and females as circles. When they produce some children, we put the line between them to indicate the mating has taken place, and then we have a little tree branching down for all the kids in the family that are listed in order of birth. So in this order, first, second, third, etc. Twins that are not identical, they are dizygotic. They are not any more related to each other genetically than these 2 siblings, but they were born at the same time, and so they're just shown this way. So it would be, there'd be some kids, and then there'd be your dizygotic twins shown like that. Monozygotic twins are a single embryo that divided because the cells didn't hold together quite right, and both of those clumps of cells made an individual. So these individuals have the same DNA. And so therefore they're shown as twins, but the line in between indicates that they're identical twins. And of course, they're always shown as the same sex, because when we talk about sex, we're talking about the chromosomes, XY and XX. For very big pedigrees, which you're probably not going to see, but you might, somewhere, a bunch of unaffected children, they often just list them this way. They because there's not enough room to put them all in. So, you might have an affected daughter, and then you might have 3 unaffected daughters, and you might have 2 affected sons, and they'd each be shown individually. But then the 2 sons that are not affected would just be listed this way, right? Individuals who are affected, the word is affected, not infected. If I see infected, I'm going to mark it wrong because I see lots of people use it, and I know it sounds similar, but it's not the same. Infected is bacteria, viruses, and fungi. Affected is when you're showing the symptoms of a genetic condition. Okay, and it's just, that's the scientific way of using the term. Some people say afflicted, but it's not really appropriate to say afflicted, because sometimes we're talking about the ability to roll your tongue or not when we're showing inheritance of these traits. Excuse me. And so rolling your tongue is not an affliction. It's just a condition or a trait or an ability. Whether your earlobes are attached or not an affliction. So this is the correct scientific term, and we color in the box, the square, or the or the circle. If you're heterozygous, excuse me, for an autosomal recessive trait, we say you're a carrier, and we color it in this way. And if it's sex-linked recessive, you can only be heterozygous if you're a female, and it's shown in a different way so that it distinguishes. But you're never going to see these on any questions I ask you, because I'll be asking you that information. Who's heterozygous and who is a carrier, and so on. Individuals that are deceased, for whatever reason, there's a line through them. I think I'll just skip over this because I don't think it's going to be relevant to anything we cover. The person who first comes to the attention of the medical people is called the propositus, and then there's an arrow that indicates them. And so this is how we draw the pedigree. Each generation is indicated by a set of Roman numerals, like that. And when we're describing something, for example, this is individual 1-2, who is the mother, and it's the trait, whatever it is, has been passed on to individual 2-2, right? And if this was an X-linked dominant trait, for example, then we would see that passing from the mother to the son. But if you were describing it, we'd want you to say, the individuals, identify them that way, okay? And if there were further offspring, you would, in subsequent generations, they'd all be labeled that way and identified that way. And this is a consanguineous marriage. This means these 2 people are related to each other, probably cousins or second cousins. That means they have either grandparents or great-grandparents in common. Therefore, they are more likely to both be carrying the same recessive alleles. So geneticists love consanguineous marriages. They are not a good idea genetically if your family has a lot of particular conditions, because you're much more likely to have a child with that condition married to a close relative. But for geneticists, like some rare traits we almost never see unless there's a consanguineous marriage. And so that's our only opportunity to learn about it, and even learn things about how to treat it and so on. So you will see in some examples the double line there. Okay, so autosomal traits. And if you have your notes, you might want to scribble in the genotypes for these. The kinds of things that make us certain about autosomal recessive. Recessive, in any case, is this, which is you've got 2 parents who are both unaffected, but they have had a child that has the condition. And that's classic, recessive. It's not the only way we can identify a recessive trait, but if we see this, we know for sure, right? And we always like to be sure if we can be. And the other thing that, in this case, we know the exact mode of transmission. It's not most likely, it is the mode of transmission, and that is it must be autosomal because we have an affected daughter, but her father doesn't have it, right? So if she had a condition that was X-linked, both of her X's would have to have the recessive alleles on it. That's how you would write her genotype. But her father would have to have given her one of those alleles. And so he should also be affected. If he's not, that means this can't possibly be the correct mode. It must be an autosomal trait because both parents are heterozygous, and now they can both pass that recessive allele on to their child. Okay. Autosomal dominant traits. In any kind of dominant trait, if you've got an individual who has the condition and you have their information about their parents, one or the other of their parents must have it, right? Both don't have to have it, but one or the other should have it. And there's no sex specificity, just like in autosomal recessive, in autosomal dominant, there's no preferential passing from one sex to the other. None of that crisscross inheritance. So in this case, we have a male who has the condition. He has one affected parent, and it must be, because it's male to male transmission, it must be autosomal. It can't be the X chromosome, and it couldn't be the Y if you were thinking of the Y, because this male is not affected. All the males would have to have it. So fathers pass on to daughters and sons with equal frequency. If this was the other way around, the mother could be affected and the father not affected, and she would pass on that allele to her daughters and sons with equal frequency, equal probability. Okay, so autosomal is the hardest one. I will show you at the end of the lecture a method to I think most efficiently determine the most likely mode of segregation. Okay, so things to watch for. The affected offspring can have unaffected parents, and there's no sex specificity in how it's passed on. Affected offspring for autosomal dominant must have at least one affected parent. If the parents are not showing it, they're not going to have that trait in any of their children, and there is no sex specificity in how it's passed on. And I'm going to stop there, and we'll move on to X-linked traits in the next recording.